spiral double helix, over 1 million nucleotides in length. An enzyme comes along and separates the nitrogen-based pairs, breaks the hydrogen bonds between the nitrogen-based pairs, so that you have the legs of the ladder hanging separate from each other, and half of the nitrogen-based pairs on one leg and the other half on the other leg. So where there used to be a pair CG, there is only a C on one side and a G on the opposite side. Like so. And so forth. It unzips the whole molecule. I'm only showing the beginning process here, but it unzips the whole molecule. Also present in the nucleus at this time, the cell prepares for DNA replication by having available. This is a nucleotide here. Sugar phosphate, nitrogen-based thymine. That's a nucleotide. I am going to draw this in shorthand, like so. So instead of writing the letter S and the letter P, I'm just going to draw an angle, an elbow, and have the thymine. So now I have an adenine nucleotide, that's a cytosine, that's a guanine, that's another guanine, and that's a thymine. Now that I have a shorthand, I could fill the board with nucleotides very easily. These are raw materials. These are raw materials for DNA replication. These are present in the nucleus before the DNA replication begins. And they are from your food. Big Mac, chocolate shake, large fries, Diet Coke, eat something for lunch. And this comes from the lettuce and the kangaroo meat in the Big Mac. All that, the bread, all that stuff has nucleic acids in it. Because there's cells in your food, in the nucleus of the cells, there's DNA. And it's all interchangeable parts. And we all know, if you match DNA with DNA, nucleotides, A, C, G, T, the pairings will be T with A, G with C, like so. That will be the matching. Straight line letters go together, curved line letters go together. So, if any of these nucleotides were to bump into the thymine-containing nucleotide on the leg of the DNA ladder that's separate, the only one that would stick is a nucleotide containing adenine. All the rest wouldn't fit. And the only nucleotide is one containing guanine that would match up with a cytosine hanging off of the leg of the DNA ladder. The only one matching up with adenine would be thymine-containing nucleotide. And when they line up like that, you notice the sugar and the phosphate on the other side, the sugar and the phosphate line up, where the sugar is in the vicinity of the phosphate of the adjoining, of the neighboring nucleotide. And what happens is another enzyme comes along, DNA polymerase, comes along and catalyzes the attachment of the sugar and the phosphate in a covalent bond. So where you had originally a DNA molecule, I'm making this up, where you had originally a section of DNA molecule that had this set of nitrogen-based pair, that genetic code, upon separation, complementary pairings are removed from each other. But because the matching of nucleotides is specific to this pairing. You can only have adenine here, you can only have thymine there. You can only have cytosine and guanine. This is a faithful copy, a reproduction of this genetic code. It is. That's a faithful copy of that. And on the other side, A, T, T, A, C, C, G, A. Now you have two DNA molecules with the genetic code faithfully reproduced. And the copying is specific and particular. The copying is exact, consistent, because these pairings are the only ones possible. Questions on this? That's a DNA replication. You have one in the beginning, two legs separate, nucleotides come around and match up. Then you have two DNA molecules, DNA replication. No. And before I go on, we're going to do protein synthesis next. Before I go on, I want to do something else. There is such a thing as purines. Those of you reading the book over the weekend, you may have come across this, purines and pyrimidines. These are chemical classifications, families of organic molecules. Purines and pyrimidines are cytosine, adenine, thymine, uracil, guanine, nitrogen bases. Families of organic molecules. What is that? That is a cut pyramid. Pyramid has been sliced across right to left. Transverse section pyramid, cut pyramid. If you can picture that in your mind, you're a visual learner like I am. I remember that picture. And this tells me cytosine, uracil, thymine. The three letters that make up the word cut. These are pyrimidines. These are the pyrimidines. If you can remember cut pyramid, then you know cytosine, uracil, thymine. And the ones that are not included, adenine, guanine, adenine and guanine are the purines. So you can remember which is which, if you can remember that visual. Okay. DNA replication, protein synthesis. Protein synthesis is a similar process. So DNA replication is a process to help you understand and remember protein synthesis. Protein synthesis is more complicated. Protein synthesis occurs in two steps. The first step is transcription. Transcription, to transcribe, is to copy. Like when a doctor gives notes, talks on a dictaphone, a recorder, and then the clerk, med assistant, stenographer, listens to the recording and types it out. Transcribes from voice to written. It's transcribed. Transcription is to copy. Transcribe would be from DNA genetic code to RNA. From the DNA genetic code, messenger RNA is generated. Messenger, mRNA. The M stands for messenger. I think you can all spell messenger. I don't have to write that out for you. So DNA genetic code is copied as messenger RNA molecules.